Technology benefits us all. Microprocessors, magnetic resonance, and powerful transmitters have changed our day-to-day lives, but not on their own. The value achieved by new technologies is when they're part of a system that provides a needed solution. Computers, MRIs, and cell phones are integrated systems that provide users with a solution. Vantix patented technology is impressive, but the true value is the integrated system that puts diagnostic information where it's needed at the speed of now.
Biosensors will transform the way healthcare is delivered by bringing rapid diagnostics to the doctor's office.
It produces the results so quickly because of the measuring system we use. It's an electrical output signal. So we're able to combine that with simple biochemistry to produce a very rapid electrical output on the system.
The electrochemical technology that we use is a potentiometric method, and this means it allows us to be able to do many different types and formats of assays at the same time on the same sample in the same flow cell. So we actually do immunoassays, enzyme assays, or sometimes even DNA assays. That means we can measure many different parameters of a disease and give a much better diagnostic.
Well, the sensor format itself, we use screen printing to manufacture it. So if we need to make more sensors in a row for one sample, we can relatively simply do that. But also we can change the biochemistry that's going onto the sensors very quickly. In a lot of cases, a lot of assays are antibody-based, so it's a matter of applying a different antibody specific to the thing you want to test. So that means that we can use reagents that are currently out there, so say antibodies that are specific to tests, and apply that rapidly onto our system. So it's a matter of transferring those antibodies, for example, onto our system and optimizing the test.
In this building, we produce the sensors for use on our point-of-care systems and our research systems. The production of sensors has been investigated to make sure it's a robust process, so we're constantly producing the same sensors each time to the same standard with good quality. We've used a variety of techniques, especially Six Sigma techniques, which is an industry-accepted standard for developing production lines and manufacturing processes, so that we can assure our customers and other teams that they're getting the same product day after day. The screen printing process is similar to commercial processes used elsewhere. We use medical grade equipment downstairs to screen print the sensors. We have a large production capacity and it's very flexible. We produce a variety of sensor types using the same equipment. During the polymerization, we apply a conducting polymer onto the surface of the sensors, and this adds value by making them much more reproducible, much more sensitive, so that when we put the other reagents on top, they will react in a much more controlled way to the analyte being tested. In the biodot process, we apply the biochemicals onto the surface of the sensor. These biochemicals will respond to the analyte that we're wanting to test for. The biodot will very reproducibly apply a specific amount of reagent to each electrode on the sensor. These are then dried, treated before the sensors are cut up into individual units for use in the cartridges. We produce different formats of sensors. The process is flexible so that we can produce one format for handheld, another format for point of care. We've also developed experimental types for evaluation and for special applications.
The VR2 is an open access 12-channel device that is applicable to many assay formats. The device consists of 12 sensor cones for developing multiplexed assays, easy-to-use reader system with control software, USB connection, battery or USB powered. The portable device provides results in minutes versus hours for currently used reference lab ELISA assays. To run an assay, first we prepare the reagents and biosensors, then insert the sensor comb into the clip, turn on Vantix VR2 reader and start the software. Select the protocol you'd like to run. Incubate the sensors in your sample matrices, wash the sensors and incubate in the detection antibody conjugate. Next, wash and attach the clip into the Vantix VR2 read head. The final step is to immerse the sensors in Vantix Enhancer solution and immediately start reading results. Once the run finishes, save the results for further analysis.
Well, the first product we're developing is the point-of-care system where we're measuring thyroid hormones. The two hormones we're measuring are free T4 and TSH. When we run these samples through clinical analyzers and compare them to our system, we get the same results. We get the same equivalence in performance, we get the same levels of precision. The way it works is that we take a small sample of blood from a patient sample. It's a tiny amount of blood. We load it into the cartridge first of all by touching the capillary tube within the loading device to the blood sample. We then take that and slide it into place on the cartridge, and we put that into the cartridge reading device. Now what happens once we've loaded that into the cartridge reading device is that it pulls the blood sample across the sensors and captures the things that we're trying to look at, so in our case the hormone markers. The system will then flow across two other solutions that ultimately results in the outcome of the assay, so the millivolt electrical signal that we get, and that is what we use to determine the concentration of the things that we're looking for in the blood. In order to move information around quickly, we've developed a web-based application called VLab. It's responsible for collating the patient information, appending the results from the assay and test together, controlling all the QC information, medical billing records, and putting it into a database that can be used by the physician.
Vantix is a privately held US company with offices outside Boston, MA and in Cambridge, UK. The US team comprises engineering, quality, regulatory, marketing and corporate. The UK team has expertise in sensor manufacturing, assay development and optimization, contract research support and biomarker development. Current priorities are distribution of the point-of-care platform in the European Union, India, Russia, China and Brazil. Co-development of markets such as retail clinics, biomarkers and veterinary. Sales and marketing of the VR2. The Vantix system is one core system with multiple commercial applications. The major market is the physician office, where the rapid provision of results can facilitate faster clinical decision-making, improved patient compliance and greater patient satisfaction, all of which can lead to improved clinical outcomes. Vantix Diagnostics, a well-established company poised for growth, has the capabilities to transform healthcare by delivering diagnostics at the speed of now.